Hi everyone, welcome back to Acing AP Biology. In this lesson, we will go through the details of membrane transport. Selective permeability indicates that the plasma membrane allows some substances to cross more easily than others. Small nonpolar molecules like carbon dioxide and oxygen can cross with ease. Small polar molecules like water can also cross easily. Ions and large polar molecules have difficulty crossing. Now let's talk about the difference between active and passive transport. Passive transport occurs when molecules move from an area of high concentration to an area of low concentration or down their concentration gradient. This process does not require the input of energy in the form of ATP. Forms of passive transport include diffusion, osmosis, and facilitated diffusion. Active transport occurs when molecules move against their concentration gradient, moving from a low concentration to a high concentration. This requires ATP. Forms of active transport include the ion pump, co-transport, and bulk transport, which can be broken down into exocytosis and endocytosis. Diffusion is the movement of a substance down its concentration gradient until dynamic equilibrium is reached. It is important to remember that dynamic equilibrium does not mean that movement stops. Molecules continue to move, but they move across the membrane at the same rate, so the concentration of molecules on both sides are equal. This process does not require ATP. Osmosis is the diffusion of water across a selectively permeable membrane. Water moves from a concentration of high free water concentration to a low free water concentration. Free water molecules mean that they are not bonded with the solute. An easy way to figure out which way water moves is that water always moves to the side with more solute, because that is the side with less free water. Remember the phrase, water follows solute. Osmotic concentration is the total solute concentration of the solution. Osmotic pressure is the amount of applied pressure needed to prevent pure water from crossing the selectively permeable membrane into the side with more solute. Or in other words, the amount of pressure necessary to inhibit osmosis. The osmotic pressure of pure water is 0. This membrane is only selectively permeable to water, not solute, so water will cross into this side during osmosis because this side has more solute. Solute concentration with respect to the cell can be hypotonic, isotonic, or hypertonic. A hypotonic solution is a solution with a lower solute concentration compared to the solute concentration inside the cell, so water moves into the cell during osmosis. An isotonic solution is a solution with equal solute concentrations, both inside and outside the cell. So water moves in and out of the cell at equal rates. A hypertonic solution is a solution with a higher concentration outside the cell compared to the solute concentration inside the cell. Water exits the cell during osmosis. Solute concentration is essential for cell viability. Since animal cells do not have cell walls, they cannot withstand large osmotic gain or losses of water. Therefore, animal cells are most healthy and stable in isotonic solutions, where there is no net movement of water because dynamic equilibrium is established. In hypotonic solutions, water moves inside the animal cell and the cell lyses or bursts. In hypertonic solutions, water moves out of the animal cell and the cell shrivels or crenates. Plant cells prefer hypotonic solutions because of turgor pressure. In hypotonic solutions, water enters the cell, specifically entering the central vacuole. The plant exerts an equal and opposite force to the water entering in, providing support to the plant and making the plant turgid. A plant cell in an isotonic solution will be flaccid or limp because water enters and exits at the same rate, so there is no turgor pressure. In hypertonic solutions, plant cells will plasmosize as water leaves the cell. The plasma membrane pulls away from the cell wall and shrivels. Facilitated diffusion is the diffusion of substances across a membrane with the help of a transport protein, which is a specific type of integrin protein. Transport proteins are specific to what solute can bind to it. It can be either a channel protein or a carrier protein. Carrier proteins switch between two formations. Once the specific solute binds to the transport protein, the shape of its binding site changes, and it releases the solute to the other side. Once a new solute binds, the shape changes once more, and the solute is secreted to the other side. Channel proteins act as selective channels specific to a certain solute. An example of facilitated diffusion is aquaporins. Water can cross the membrane on its own. However, this diffusion is too slow. Aquaporins are transport proteins that allow the crossing of water, and thus they increase the rate of diffusion. That's it for passive transport. We will go on to active transport in the next video. See you then. For more bio content, hit subscribe.